Our speaker tonight is Ryan Contreras. He is a native of. We got a few people here who from advancement who need to leave. Ryan is a native of North Carolina, Eastern North Carolina. He's now out in the Willamette Valley of Oregon. He is one of our own. He's got more than one degree from NC State, working with Tom Ranney, doing breeding work. He's now out at Oregon State University, where I have friends out there, so I hear a little bit about what's going on out there, and we'll figure this out afterward. Ryan is taking the lessons he learned from Tom pretty seriously. He's breeding everything from Abelia pescin, Trachlosperma. Boy, I'm director for a month and I'm already losing plant names. But also really inspiring a love of plants through plant materials, plant ID courses out there. So really doing some exciting work. He's outdoorsman when he isn't gardening and teaching. He's hiking and cycling and doing all kinds of things like that throughout the beautiful Willamette Valley with his family and new son. Is that right, Cooper? Relatively new. So starting on the next generation of gardeners. So please help me welcome Ryan Contreras.
Thanks, Mark. A couple of quick things. Can you guys all hear me? Is this good? So this is the first time that I've worn this tie since I've graduated, and it's also the last time that I wore this tie was the last time I spoke in this room. So when I graduated from my master's degree back in 2006, and so Richard Olson, we both work with Tom Ranney. He graduated in 2005, and when he gets up to receive his degree, say a few words, he starts crying. He was very close with JC, and he was such a mentor to him. So Richard got, choked up, and so I gave him a hard time about it. Of course, I get up in 2006, and I'm saying a few words, and of course, the waterworks start. So I'm hoping I'm not going to cry tonight. The other thing I've got to say before I forget it, Mike Worthington was in here. So I am a true NC State guy. One of the great things that they do at NC State when you're getting your degree, whether you're no matter who you're working with, you're required to do a formal extension experience. So who better to do a formal extension experience in nursery crops with than Ted Bilderback? So we were actually on the way back from Worthington Farms. We've been out there, and I'm just, to say that I learned a lot with Ted on my 3 trips it doesn't do it justice. I was on the way back, and the question that I get asked more than anything, so I'll show you I'm working on 18 or 19 general, but the question I get asked more than anything is, how do you figure out what you're going to work on? Like what's there's so many nursery crops and all that. So I asked Ted, like this, years and years ago, 10 years ago, I asked Ted, so when I get out, assuming I get my PhD and I'm go on, how do I figure out what I'm gonna work on? And Ted's advice was, well, not every yard has a cherry, not every yard has a birch or a crabapple, but damn near every yard has a hedge. So I've sort of taken Ted's advice to heart, and you'll see a lot of what I'm doing is working on hedges. Okay, so anyway, I'm trying to convince you that I do come from NC State. So my talk today, I didn't really know what I was going to speak about. I didn't want to show you guys a lot of data. I'm an academic. Data is like a warm blanket to me, so I've got to show you a little bit of data. But I really want to tell you okay. So I want to tell you a story. I've had the opportunity to work at some of the most amazing institutions in the country. I've had a chance to meet some of the most amazing plantswomen in the world. And so I'm just going to talk about some of my experiences from NC State down to Georgia and then out to Oregon. And so I had to throw in the subtitle. I'm a big barbecue guy, so from barbecue and grits to salmon and quinoa, and I assure you I've been there for 5 years, and you cannot find Eastern North Carolina barbecue outside of my house. So I'm from Beaufort, North Carolina. If I have a couple of bourbons or talk to somebody from back home, that does come out a little bit in my accent. So I spend a lot of time doing this. I don't have one of those amazing stories of like, my grandfather or my grandmother really instilled a passion. I spent more time over at Shackford Banks and over at Cape Lookout than I did gardening. We did have 25 acres of coastal Bermuda hay that I grew up slinging 60-pound bales, though you wouldn't know it to look at me. But I did eat a lot of Eastern North Carolina barbecue, and if you're wondering, I am a Wilbur's man, although I eat King's on my way home, and my God, it does not disappoint. So I took my son Cooper to Clyde Cooper's earlier today, by the way.
cool.
Just today I ate at Clyde Cooper's for lunch and K&W for dinner. My God. So it's good to be back. Sweet tea. So I came to NC State in 1996. I got my bachelor's in horticultural science, and just the straight track, I wanted to be a nursery grower. Right, and so I was not always the put-together specimen that you see before you. If you knew me. Back in the day, I was a little bit different, and I was working at Tuning Nursery, which is out in Apex, grows a lot of azaleas, and so I got cured of wanting to be a grower when it was August, 95 degrees out, and Charles, the owner of the nursery, Charles Tuning, he took a week and went down to the beach with his wife, played in volleyball tournaments, and went on vacation for a week. And it struck me one day when I'm out tinkering with the irrigation that I was the only thing between complete failure of his livelihood. And so I became cured pretty quickly of wanting to be tied to the nursery. At any rate, going through, so as I was finishing my degree, my last term I did an independent study project with Danny Werner, another one of our previous directors here at the JCRA. And we were, Denny is obviously, he's been doing some really breakthrough stuff on Buddleia, but back then he was interested in Buddleia weyeriana and honeycomb, and so this is what we wanted to do. Basically, he wanted me to count the chromosomes of Buddleia honeycomb and figure out how many chromosomes it had, and if that was what was potentially impacting its fertility at the time. So this is what we want to do. Again, this is not numbers data, but this is one of my photomicrographs. This is actually Calocarpa japonica leucocarpa. So now you can tell everyone that you've seen, you know what the chromosomes of white-berried Japanese Calocarpa looks like. You're amazing. So, he put me in touch with, this is Dr. Sham Tallery. He was in peanut genetics at the time here at NC State. He's now the peanut breeder for Clemson. We lost him to Clemson. But he is an amazing cytogeneticist, and so he really did teach me a lot of what I know, such as it is. So, amazing cytogeneticist. So I went out and worked in peanut genetics. Doesn't that really get your blood pumping, your heart racing, looking at that, how gorgeous that is? So that was, spent a lot of time down in Lewiston. Amazing. So this is some of the things I learned with Sham was, cytology, which is looking at chromosomes. We worked with molecular markers. I got to do a lot of controlled crosses, pollen staining and germination, doing some lab techniques like that. Histology, which is preparing slides with very thin sections, and we stain different tissues. Breeding schemes, and that, how to go about the process of developing a strategy, going through the breeding schemes, and then seed handling. Really quick story. I worked for 2 years in the peanut genetics program. One of the things that they do, and they still do, is they maintain the wild species collections of peanuts. And so that's down in the eastern part of the state, so they go periodically and plant out all of these, hundreds of species, and they are dug, they are put into flats, and so you come back with a big pile of dirt and rocks and peach pits, because it's down at Sand Hills, and all of this stuff. And so then you have to filter through and get out these little tiny peanuts. Right, because they're much, much smaller than cultivated peanuts, that we're used to seeing. The first year I did it, I kept saying, there has got to be a better way than sitting here and going through and picking through like, a monkey picking lice off of the back of one of its partners, right? And there's got to be a better way. And I kept being told by the technician who had been there for, decades that this is how we've always done it. All right, that's how we've always done it. I guess I'm going to sit here and I'm going to listen to you, and we're going to do it. Well, she got promoted or transferred to the peanut breeding program, so there was no technician ahead of me in the peanut genetics program. So the next year, I went to every seed breeding program at NC State, and I asked them, how do you clean your seed? Can I see your equipment? Can I borrow it? I got my hands on this thing from the 50s. It was called a clipper. They make a new one, but this one was so old it had a leather belt, is what ran this rig. So what took me about 8 months, with 3 of us the year before, I did by myself in about 6 weeks. And so after that I said, you know what, I cannot spend my entire career doing things the way we've always done it just because that's the way we've done it. I'm going to make my own mistakes, and I make a lot of them, but they're going to be mine. So innovation in seed handling is what I taught myself. But lo and behold, and as much as I learned and as much as I loved the people that I was working with and the free peanuts, I ate a lot of peanuts. This was, it drove me batty. I can't stand walking into a field of peanuts. There's no diversity. It does not get me excited. This is what gets me excited, right? So I had to come back to the glorious world of ornamental horticulture, right outside our JCRA. I can't believe that flipped through. I love this photo. I was over here in the West Garden with an old friend and graduate student, Brian Krug, and we were taking a look at here. That's the most unique fruit I've ever seen. Anyway, I had another quick story. I'm never going to get through all these slides, but I got to tell you. So I feel so gypped in my horticulture career because I literally showed up right as JC passed. I mean, there was a memorial was going on in the West Garden the very first time that I came to the North Carolina State Arboretum. So anyway, I was just that close to meeting one of the greatest plantsmen ever lived. But another great plantsman, another great mentor, Tom Ranney. This actually happens to be Iron Mountain out in Oregon. He came this summer and we did a little hiking after the Far West show. So I came back to ornamental horticulture. Tom took me on in spite of a D in quantitative chemistry that I still think about. I was also 10 minutes late to the first time that I met with him, and Denny looks at me and he's like, "What are you doing? You're meeting with Tom Ranney and you're 10, 15 minutes late?" Anyway, like I said, I wasn't always the put-together specimen you see before you. So I spent my summers doing research up at the Mountain Horticultural Crops Research and Extension Center with Tom. I put Fletcher up here. It was then Fletcher. They've now redrawn the lines and it's Mills River. But anyway, a really amazing place to spend your summers. I worked on azalea dendrons, and so people think that's a made-up term, but it is actually a cross between a deciduous azalea and an evergreen rhododendron. And in fact, the first documented cross, or documented hybrid in the genus rhododendron was called Azaleoides. So it actually was one of these azalea dendron hybrids. Well, some of the work that we did was to confirm that it was actually Rhododendron ponicum crossed with viscosum. It was fragrant affinity. It does tend to combine fragrance that we get from swamp azalea and evergreen foliage from ponicum. And realistically, it's actually sort of semi-evergreen. The leaves will die and they kind of hang on the plant, so it's not super great. So it's not an endpoint. I mean, we wanted to use it as a breeding program, but unfortunately, it's got this hybrid sterility. So think about it, you cross a female horse with a donkey and you get a mule, and if that mule's not what you want, it doesn't matter because that's the end of it, right? So mules are sterile. So that's what we had in rhododendron fragrant affinity. So we wanted to, and again, this is rhododendron ponicum, viscosum, and there's fragrant affinity. You can see it is a nice hybrid of the two. So what we wanted to do was to use it in a breeding program. I'm going to talk about induced polyploidy here for just a minute. And so just a couple of definitions here. Ploidy is simply the number of sets of chromosomes that a plant has, right? So we're all diploids. We have 2 sets of 23 chromosomes, so we have a total of 46 chromosomes inside our cells, so we are diploids. Anything above diploids, so triploids, tetraploids, pentaploids, hexaploids, so on, are all what we call polyploids. Okay, so why does that matter, right? So we take advantage of induced polyploidy, or doubling the chromosomes of fragrant affinity, and that actually can restore fertility. So if you have, this is sort of what we have in rhododendron fragrant affinity. If you think this is rhododendron ponicum chromosomes, or their genome, and this is viscosum, they're too dissimilar, right? So they don't pair up during meiosis, and what you get is sterile pollen, sterile eggs, right? So it just doesn't work. It's a dead end. But if you double the chromosomes, you can actually provide a partner for each chromosome to pair with, and it restores fertility, and then we get fertile pollen here. So this technique was really popularized. It was going on before this, but this was the seminal paper by Blakesley and Avery back in 1937. What we're doing, there's been a resurgence in the last 20 years in ploidy manipulation, but we're just going back to what people have already been doing. Newton said, "If I can see further, it's from standing on the shoulders of giants," and that's all we're doing here. So they were using colchicine back then, and this is a really fantastic paper. If for no other reason, the photos are incredible. Okay, so this is some of the different techniques that they use to double chromosomes, right? So this has got colchicine. Everywhere you see a test tube here, it's got colchicine in it. This is portulaca. It's So they're like, you can bend it over and let the meristem grow down in there. You can flip the pot over. I don't know what they had on there. I'm kind of fascinated. I'd love to see inside that pot. But they've got these steaks and they've flipped it over. This is just a lot of work to make. You can use glass wool or cotton soaked in colchicine and let the meristem grow up in there. You can take in, this is wicking out. So this is a colchicine solution, they're wicking it out to get it to the meristem. This is by far my favorite. That is a jug of colchicine. Colchicine is not good for you folks, okay? It's really, really bad, okay? And so he's got this atomizer. So he's atomizing, making a very fine mist of colchicine, and you can see his personal protective equipment. He has a tie. But I do love that, doing science with a tie on. There's just something I really feel good when I've got a tie on at work and I put a lab coat on. God, nothing like it. Anyway, and then there's a little dropper being added here. The bottom line is, it doesn't matter, the common thread is you've got to get whatever the anti-mitotic agent is, in this case colchicine, onto the meristem. So that's whatever the effective way of getting it on the meristem is. And that's it. So we were successful in doubling the chromosomes of fragrant affinity. So this is what the diploid looks like on the right, and the tetraploid over here on the left, flowers roughly twice the size. So in addition to restoring fertility that I'm going to talk about in a minute, we made just a better plant. Flowers twice the size. Everybody can see the benefit of that. So this is pollen from the diploid. It did not germinate. We put it in a nutrient media, look at it on a microscope. It does not germinate. We tried to stain it with acetocarmine. It does not stain. You double the chromosomes, pollen stains, pollen germinates, voila. Well, so so staining in the diploid, very low, staining very high in the tetraploid, and pollen germination, almost to 50%. So going from 0 to 50, pretty good. Then we made a bunch of crosses with fragrant affinity. So we tried with the diploid, and we got no seed set, no seed set, no seed set, regardless of what we were crossing with. Incidentally, we were crossing with Catabiense because that was what was in the literature as reported to be the evergreen rhododendron parent. Our molecular work showed that it was actually Ponicum. At any rate, we can see that when we used the tetraploid fragrant affinity, we did get, it was low, but we did get some seed set and it actually germinated. So we have effectively restored fertility. There, just to show that I did used to make crosses. I have to break out these photos. My grad students don't think I actually do any science anymore. I was sitting in an office, but I did make some crosses. We crossed that restored fertility tetraploid fragrant affinity with Vulcan, tetraploid, supernova, Briggs red star, and polar bear. Anybody know polar bear? This is a really fantastic. I'm big on fragrant plants. This has a melon fragrance. Anybody know this? I suggest you find it and grow it. I know it's hard to find, if it's in the genus Rhododendron and it's not an encore, it's kind of hard to find it anymore these days. Alright, so, see, these are some of the plants. The plants actually look great. I was really excited in the greenhouse. I was like, man, I've made, only a few thousand crosses just as a master's student, and I've got these gorgeous plants. They're really phenomenal. So here's what the fragrant affinity by Vulcan, the polar bear by fragrant affinity, supernova, I was really excited about these plants. But you put them in the field, and this is what happens. Basically, and this is late fall going into winter. And this is in Fletcher, Mills River. So they're sort of semi-evergreen. The leaves die, and they hang on, and they're not fragrant, not a lick. So it'll come back. We'll talk about fragrance and some of my work with mock orange a little later. So I was really disappointed in fragrances, and talking to Tom about some other projects. It really seems to drop out fast. So fragrance is a challenging one to breed for. So that's quickly what we did up in Fletcher with Tom. Then I was looking around. I didn't know where to go. John Reuter was actually Richard Olson worked with John Reuter down at University of Georgia on his master's. Came back, got his PhD with Tom. I got my master's with Tom, and then went to work with John Reuter at University of Georgia for my PhD. It seemed like a reasonable thing to do. He was pretty successful down there, and Reuter, he sort of recruited me to come down there. And before I went, I asked Richard, "What's the deal with Tifton? What's the town like?" He said, "You're going to get a lot of work done." I said, "Oh, I know it's not a big town, but what kind of town is it?" And he's like, No, really, you're going to get a lot of work done. Sure enough, I got a lot of work done in Tifton. Let me tell you, 64 miles above the Florida line, dead center of the state. The station where we worked was actually established for a heat tolerance station or research because it's far enough away from the Gulf and far enough away from the Atlantic. It gets no moderating effects from either of those bodies of water. Really fantastic place. Okay, just showing you, here's John Reuter, Jim Owen, who I was at, well, we were both at NC State. He was at Oregon State at the time. He's now at Virginia Tech, and of course, Richard Olson. So I feel like, so John, actually a lot like Tom, he was actually trained as a plant physiologist. So he did all this electrolyte leakage work in hollies and all of this. So he wasn't, he wasn't a traditionally trained breeder, but he knows plants. This guy is a serious plantsman. When I got down there, he just had loads of germplasm and sort of he just opened the door and said, do what you want to do. And so my eyes are much bigger than my stomach, and so I'm going to work on everything, which really was as good a training to become a professor as anything has ever been. He just let me do everything. So I got this really broad experience. I had a couple of professors at Georgia tell me that, I was making big mistakes with this level, or, doing this diverse amount of work for my doctorate, because when you get done, you should be an expert. Right? So their idea was like, you should work on cryptomeria and be an expert in cryptomeria. But my response is, there's no single crop that you could work on that can allow me to get this diverse level of training and all of these different skills and different techniques that I did working on, Calocarpa, Cryptomeria, Hibiscus, and Tacoma. So not only are they 4 different genera, 4 different families, they're as different as anything, as any plants that are out there. So I worked on lots of different stuff. I got to go play in John's plots. He is a conifer expert for the South. So I've got an old JCRA shirt that says, yeah, we can grow conifers in the South, and it's got the whole list of conifers on the back. Well, John is down zone 8, right, middle of the state. It's hotter than fire, and he tested all kinds of stuff. So in fact, that is, anybody know that pine? Jared, no? Anyone? That's Pinus willikianus, Himalayan pine growing, it averages over 100 days at or above 90 degrees. Doing fine. There's coastal redwood in there. Anyway, it was a really fun place to work. There's John. This is Calocarpa longissimum. This was one of my favorite Calocarpas. The first year that we had this thing in the ground, it grows like a madman. I mean, this was early in the year. It still had more that was going to happen. But actually, the berries stayed pearl white. They were gorgeous. Next year, not so much. I'm still excited about this plant. So John actually has released, this is Southern Moon, Rhaphilepis. It is a big improvement over Minor. I don't know how big Minor gets here, but Minor, which was a Monrovia release, was supposed to be a 4 to 5 foot tall Rhaphilepis. In Tifton, it was 8.5, 9 feet, 10 feet. This is a true low-growing dwarf Rhaphilepis. So this one's also coming out by Monrovia. And I got to, so in addition to that other stuff, I was, I was a graduate student, so it was whatever. Go harvest some tea oil, camellias, go make some crosses. So I got to play around with camellias. Again, I tell you, I'm from the South, so we worked on camellias. While I was down there, this is my one of my favorite stories. My wife, I can hear her rolling her eyes right now that I'm about to tell you guys the story about the Ashburn house. All right, so the first house that I lived in when I moved down to Tifton, this was outside of Tifton. I was cheap. I wanted to live as cheaply as possible. So this was the view outside my back door. It was all farmland around me. It was an old cinder block farmhouse. And everybody told me I was crazy for moving into this house in the middle of nowhere. It was on Maud Williams Road. That's country right there. So moved into this place, me and my dog Callie, my trusty sidekick. And so one night, I had fallen asleep on the couch watching a movie. It was a Saturday night, so I was really living it up on Maud Williams Road. I woke up, 2 o'clock in the morning, bleary-eyed, and I go into the bathroom, and there's a snake on the back of the toilet. Right? So I say that I should write a short story, and it will be called Fighting a 4-Foot Snake with a 5-Foot Shovel in a 3-foot Bathroom. This is what happened. So the best part is, that was Saturday. I go in, I see the snake, and I literally I slam the door, I run and I jump on top of my ottoman, and I'm still screaming like a small young woman, and I'm just screaming and screaming, and then I realized that, oh my God, there's a snake that I should probably go deal with that's in my bathroom. So by the time, blood comes back in my brain, I go get boots on and I grab my shovel and I go back, and of course the snake is gone at this point. So I spent the next several days in a panic. I don't like snakes very much, so I was really stressed out. I did not sleep well for several days. But by Wednesday, one of the great things about working with John is I'd always bring plants home. All right, so they'd be pulling stuff out of the pot, and I can't let it go into the the burn pile. So I'd take it home, and I was planting plants outside this farmhouse, and it started getting a little bit late, and I needed to go inside and take a shower. So I go inside, and just to be perfectly frank, I was getting ready for a shower. I don't shower in my clothes, so I was preparing for a shower, and I was all the way prepared for a shower, if you will. And so I had to use this facility. So this is a photoshopped image of a snake, but I did open the toilet, and there was a snake in the toilet. So I dropped the lid this time, as scared as I was. Boots be damned, boxers be damned. I went and I got my 5-foot shovel, and I came back, and toilet be damned. I almost broke the toilet, but I go after him. The snake comes up, like the Kraken. We ride around, and but I did slay the snake. If you're wondering, it was a corn snake, but I felt really bad. But we have this agreement that don't come inside the 4 walls, especially in the toilet. I'm sorry, but all bets are off. I love nature, but all bets are off at this point. We did have an intern that was working. This is not her. It's from the internet, but we had an intern working with us, and I had it in a can with me because I was going to get out of that lease. So I took the snake and I showed it to my landlord, and he let me out of the lease because I was like, there's no going back. There was one toilet in that house. I'm never sitting on it again. I'm sorry. That toilet is done. It's dead. It's dead to me. But this intern, she says the same thing I know some of you were thinking is, you didn't have to kill the snake. And I just, again, you missed the part of the story where I told you there was a snake in my toilet. You missed that part. So anyway. All right. So in addition to that excitement, I did, we went on some really amazing trips down. One of the best, and I'll never forget it, was a trip down the Gulf Coast. John and I, there's Dr. Reuter again, John Reuter, we hopped in the truck and went for about 10 days all over the Gulf Coast. One of our stops was Buddy Lee. Some of you may know Buddy. He's the developer of the Encore Azalea, among many other things, now with PDSI. There's never been a more unassuming guy than Buddy Lee to be as famous as he is, and he'll tell you, I've been in a room several times when he just tells everybody, here's how if you want to go develop your own Encore, here, go do it. And he'll tell you exactly what plants he used and how he went about it. So anyway, really amazing going down and seeing, there's probably 5-6 more cultivars just in this little block of stuff at Buddy's house. Really, really fantastic guy. We went down to Flowerwood here. There's Jerry Pittman. Got to hang out with those folks, and they're doing nursery production on a fairly grandiose scale down there. Anybody heard of John Thornton? John Thornton, really incredible rhododendron breeder. He's down in Louisiana. It's hotter than fire. It's humid. Mosquitoes about that big, and he's growing evergreen rhodies. You'd never think. And you know how he plants them? He sets them on top of the ground, doesn't dig a hole, sets them on top of the ground and mounds up to them. That's how, he killed a lot of them. Now, Tony said you can't know the hardiness of a plant till you kill it 3 times. I imagine Dr. Thornton killed a right good many. Guy's a dentist, by the way, trained as a dentist, retired as a breakthrough rhodie breeder, breeding for root rot resistance, and I'll never think of the species right now. It'll come to me. Hyperethrum, that's what he's using. Again, another one of those guys, he'll tell you every species that he's using, every cross he's made, and everything else. So some really incredible plants. Here's one of my favorites. There's Dr. Thornton. I think that was one of his favorites here. But just a real quiet, real amazing guy. Margie Jenkins, of course, Miss Margie. No trip to the Gulf Coast is complete without going to see Miss Margie. Again, just a complete legend. I was in awe of her plant knowledge. She has, not that she forgets much, but she's certainly forgotten more than I'll ever know. Just, you know, it was just so great to get down there and see her at the nursery. And I mean, the collective plant knowledge in this photo, Buddy Lee, John Reuter, Miss Margie, and Dr. Thornton is kind of mind-blowing. So I just so lucky to get to go down on that trip. And some of you may know Tommy Dodd is of Dodd and Dodd Nursery. I forget, I'm embarrassed to forget Tommy's wife's name, but we went down, pardon?
Later.
Thank you, there. And so we went down and hung out with those folks. If you don't know Tommy, he is a really great guy. One of my regrets is that I've never been to the Cosmic event yet. That is a party that they throw, and I mean, if the name is, you know, suggests anything, I've got to go to that. I might revert to that old, not so well put together days when I'm at the Cosmic event. At any rate, they were really fantastic. We walked around the nursery, saw, you know, talked plants a lot with them. Here's Shady Lady. He loves to talk about Shady Lady. He named it for Thayer, for his wife. Anyway, that's a variegated Elysium there that he found. But we also went down the Escatawpa River and got out. He's, you know, huge into native plants from that region down in Louisiana and excuse me, Alabama, and just such a good time. And I did go off. We saw a rope swing. So am I not going to go off a rope swing on the Escatawpa River? I mean, when am I going to be here again? That's embarrassing. I'm sorry. I shouldn't show you guys that. but okay, back to a little bit more research that we did down in Georgia. So I did work in Calicarpa. I think they're just really fantastic. They need a little more breeding, but I think they should be used a lot more in the landscape. So I did some research to determine how fruit color was inherited. So I'm big on, you know, learning how traits are inherited. I don't think you can have a breeding program if you sort of willy-nilly making crosses with this and that just to see what you get. Of course, I mean, there is a place for that, there's a time for that. But I like to know how they're inherited, so we did a bunch of crosses. There's a bunch of numbers and letters and things, but crossing Calicarpa, man, the flowers are small, and it was tedious. And did I mention it's hot in Tifton? Man, those were hot days. I would spend about 6 hours crossing those rigs. But we got our seed set, and so we did various crosses with reciprocal crosses between purple and white. We grew out a lot of progeny, and basically what, at the end of the day, we have confirmed that there's one gene that controls fruit color, and actually fruit color, flower color, and petiole color in American beautyberry. And it has 3 alleles. The purple allele is dominant to the pink allele, and that is dominant to the white allele. So if you cross a purple with a white, you get 100% purple. Cross a purple with a pink, you get 100% purple. And you cross a pink with a white, you get 100% pink. That's what we found in our crosses. So it's incredible how much work went into telling you that one sentence. Really, it just all distills out to that right there. You know? It's just kind of amazing. So this is a plant. It's funny, you know, John is as straight-laced guy as you ever want to meet. But he does have a great sense of humor. So he worked a lot with Hibiscus acetosella, and he selected this one and called it Panama Red. Of course, it looks like marijuana. Panama Red is sort of a name that, you know, is reminiscent of that. So Panama Red, it was a great plant. You know, everybody liked it. It did really well in the UGA trial program. But the one thing, or a couple things everybody said about it, it just got too big. And down in Georgia, by August, it'd be 8 feet tall and flopping. So what we wanted was something that would basically stay a little bit smaller, perhaps grow a little bit slower, and wouldn't lodge, wouldn't flop over at the end of the year. So we went back to doubling the chromosomes. Some of you heard from me with this technique. You may say, well, when you double chromosomes, plants get bigger. You got, you know, more chromosomes, they get bigger. Well, actually, what often happens is just the opposite. They actually tend to stay a little bit smaller. So we did successfully double the chromosomes of Panama Red, which is on the right, and we got this plant. So the plant is overall, it's smaller, the canopy volume is smaller, the internodes are about half as long. Now the leaves actually are smaller, and I don't know if you can tell how well you can tell from the photo, but there's no green in this one. There's a little bit of green in Panama Red, but the plant is more open. You can see this guy is really compact and dense, and it is purple. I want to call this Panama purple, but it's not really gone anywhere yet. But it is compact, it's non-lodging, so it doesn't flop over. And an added benefit here is that this one is actually highly sterile. We've never gotten a single seed to set on it, at least so far. We never say complete sterility. Anytime you work in, I don't trust anybody who works with biological systems and says always or never, right? Because they find a way. Nature finds a way. But down in, if you're growing these in South Florida, actually Hibiscus acetosella has the potential to be extremely weedy. It produces a lot of seed, so this is an added benefit. And I'm still I obviously don't know the nursery industry as well as I think I do, or else this thing would already be out there and be famous. All right, so showing this photo again, in 2008, while Jim was at Oregon State, he was helping coordinate some of the education sessions at the Far West Show, and lo and behold, he got myself and Dr. Reuter and then Dr. Olson invited out to the Far West Show. So while we were out there, we went and visited Monrovia. If you've never been to the Dayton, Oregon, Monrovia location, you should go. Their facility, like you pull up to it and you think you're at a ski chalet in Switzerland. It's incredible. Soft music playing. Would you care for a beverage? Doesn't feel like you're at a nursery. At any rate, so there I met Nicholas Stadden. He is the director of new product development for Monrovia, and one of the great relationships that I still carry on today is we work closely with Nicholas, and Monrovia trials a lot of the plants that we're developing. Really a fantastic character, not a nicer guy out there. While we were at Monrovia, this is one of those great things. This is like perhaps the one time in my life that I can ever say that we were maybe a step ahead of Dan Hinckley and Monrovia, right? So Dan Hinckley was doing plant collecting for Monrovia, and so we brought back Calocarpa formosana. So one of the things I didn't talk about, we had a pretty large, diverse collection of Calocarpa down in Tifton with John, and I crossed everything with everything. If you've ever seen the spider web of camellia crosses that Dr. Ackerman did, that's what I was trying to replicate. Unfortunately, nothing crosses with anything in Calocarpa. The only things that cross are Calocarpa americana and Calocarpa acuminata, which I actually don't think is a good species. At any rate, so we were already crossing Formosana when Monrovia still had these little seedlings, so I kind of walked out there a little puffed up. I was ahead of Monrovia. At any rate, went to Hoyt Arboretum, another fantastic place. I still maintain a relationship with the curator there, Martin Nicholson, really fantastic. They actually have our Cotoneaster collection growing at the Hoyt Arboretum. Really fantastic place. We got to go to all kinds of amazing nurseries. I had to throw these in here. It was the first time I had ever seen these massive grow boxes growing really serious specimens, right? That happened to be, this is at Buckholz and Buckholz Nursery. This is also at Buckholz and Buckholz. One of the compatriots from the South, this is Tom Cox. If you've not been down to Cox Arboretum outside of Atlanta, another amazing, fantastic plantsman. And that place, it's incredible. I mean, I don't know how he keeps the weeds down as big as that place is. And actually, I didn't mention John and Tom have written a book. I can't remember it off the top of my head, but it's about conifers for the South. Anyway, more conifers. This is at Larry Stanley's place. If you want a cultivar of a conifer, you go see Larry Stanley. He is a cultivar man. He's got a zillion of them. And I keep going on with that trip. It's fantastic. So then I got the job at Oregon State. They advertised this job. I've been there since I started December 2009. I am actually, it's incredible to think this, but I'm the first person at Oregon State University that my sole research responsibility is ornamental plant breeding. So, you know, at its peak, it was almost $1 billion wholesale value, farm gate value. And to think that we didn't have a breeder that was serving that industry is pretty crazy. But at any rate, and loaded up the car, me and Callie headed west. I did see, actually my family, Contreras is not an Eastern North Carolina name. So I've got family. My family's all from San Diego, so I shouldn't have been too surprised. But this is poinsettia, right? You know, it's basically a small tree down in its native habitat. But this was really cool to see when I was out driving. Drove up the Pacific Coast Highway. This is, the 101, just really quite picturesque. And again, coming from the beach, you know, when you go to the coast in Oregon, that's what you say, I'm going to the coast, right? We go to the beach here. They go to the coast in Newport. And it's where we are. Anyway, more coastline. I'm going to show you guys. You guys want to see some plant photos. I lost my dog a year ago. Pulling at your heartstrings. I do like golf. I'm a terrible golfer, but I do love golf, and so we had to stop by Pebble Beach. And this is the 18. This is the but it is plants, right? We're talking plants. This is the most photographed tree at Pebble Beach. So by God, I was not going to leave Pebble Beach without taking a photo of said tree. Lovely trip. Of course, Golden Gate, got some plants in there. I had nobody else with me, so we did finally, eventually we made it to Oregon. Right? And so I am in Corvallis, Oregon. I have a 40% teaching appointment, and as Mark alluded to, I don't know how many of you are really familiar with Paul Funtz, but I took my Plant ID classes from Paul Funtz, and I am no Paul Funtz, but I do everything in my power to take that same level of professionalism and raising the nomenclature, and the students say, why do I have to know how to spell this? What, and it's like, because we're raising the bar, we're raising the level of professionalism as you take it out to the nursery. Anyway, I'm a big fan of Dr. Funtz. So I have a 60% research appointment, 40% teaching. I teach Plant ID and growth and development there at Oregon. But the greatest thing is, in addition to the students, which are fantastic, the greatest thing is I get to go when I go to nurseries. This is the kind of view I get, right? So this is the kind of nurseries that I get to go to. This is my good friend Andrew Bell is the curator at Chicago Bot Garden, right? And then we're at Isley Nursery. Again, I mean, it's like Disney World. It's perfect. There's not a hair out of place. It's incredible. That's another shot from Isley. What can you say? That's not like going to work. I don't care if you've got to haul pots all day. That's your view. It's still, I have a song in my heart. And this is from J. Frank Schmidt, where I was looking at maples. And again, that's not too bad. I think I could deal with that. I did meet my wife out there. This is out Bull of the Woods wilderness area. I can't explain to you how tired I was. This was on day 3.5, 4 of this hiking trip, and she walked me into the dirt. But anyway, this was really fantastic, and we picked up another friend here. This is Henry. So this is our family, such as it was at that point. We've now expanded a little bit. We have Cooper. This is Cooper and I out hiking Iron Mountain again. Fantastic spot if you're in Oregon. You want to see wildflowers in summer, go to Iron Mountain. Lovely. There's Cooper again. I can't help myself. I had to throw these in here. And we all three of us work at go to school at Oregon State every day. We get up and go to campus. He's in a little daycare. My wife and I both work for Oregon State. Pretty fantastic. So what do I do as far as the research program? How far over am I at this point? How much time have I got?
You're good.
Okay. About 3 minutes left? Let me know how
Want to do an hour? You got 20 minutes left?
Oh, we're good. Okay, so and this is this list is always in flux. I almost forgot vaccinium the other day, but we work on about 18 different genera, and people say, well, how do you work on all that all the time? Well, I mean, the thing is, I say that I'm working on Nyssa, but at this point, what I mean by working on Nyssa is I've got these plants planted out in the field, and we're looking at them for the next 8 years. So I mean, there's not like you go out and you work on everything every day, or even every year or 5 years. For the sake of, and I do think it's a pretty diverse list. I mean, that's because we have a diverse nursery industry, right? If you're only breeding maples, what are the shrub guys going to say? So we try to hit a little bit of everything. We try to hit on very economically important genera, hibiscus, Catsuricree, Cercidophyllum. Thuya, of course, is hugely economically important. Syringa, acers, Cotoneaster, not so much, but I hope to make a comeback. But we also hit some minor crops that either pique my interest or I think they have a low bar, right? So like ribes, that was our first release. I'll show you ribes sanguineum. Well, maybe not. Actually, I don't think I will. I don't think I have a slide, but our first release was ribes sanguineum Oregon snowflake. Right? The bar is so low in ribes. The cultivars that are out there, they're just, frankly, they're not good garden plants, right? They get tall and rank and out of sorts. So that's sort of, I guess, how I balance what do we work on that I like versus what do we work on that is just economically important. I don't get real excited about Thuya. I mean, I got to tell you, but it's like Ted said, there are zillions of them growing. You drive up and down the Willamette Valley, and you see millions of Thuya occidentalis being grown. At any rate, Moving on, this is a tiny, tiny snapshot of the diversity in Hibiscus syriacus, Althea, or Rose of Sharon. And an interesting story how we got to working on this. I was writing a paper with a colleague on techniques for developing sterile crops. So I cited Don Egolf's work when he released Diana and Aphrodite and Minerva and Helene as being sterile triploids. And I had a reviewer write me back and said, one of the comments was, that's great, but those plants are not sterile. And I have to admit that I had never grown them in research plots or in a garden or anything. I didn't know that they produce seed. So that set me out, and I said, well, let's get them in. Let's get a bunch of different cultivars. Let's quantify the fertility. And then I started getting into, well, nobody has ever looked at how any of these traits are inherited. And you've got, quite a lot of diversity of traits. So we've got different flower colors. There's presence or absence of eye spot. You've got, double flower, singles, anemone. And actually, if you tally it up, there are 30 trait combinations that are possible. So you got, and roughly 5, and we're siloing a little bit, right? So how do you go from blue to lavender, right? There's a lot of gradation, but we do try to put things in silos. There are 5 different flower colors, 3 what we call petal types, so you can have single flowers, anemones, which are, they have a few petaloid stamens, and then all the way to a double flower. And then you can either have an eye spot or not have an eye spot. So we between 3 years, 2012 to 14, we've made 80 different cross combinations between different cultivars. Grow out about 1,280 seedlings that we're looking at for these traits. And and I say I love the royal we. We made these crosses and we potted all these up. That's my research assistant Mara. I have to admit my hands touched probably very few of these seedlings. So I'm just going to run through. We don't have this all figured out, and I'll show you here in a little bit why we don't have it all figured out at this point. But I just want to run through some of our cross combinations, and you've maybe noticed the flurry of Althea cultivars that have come out, and it's because there's a lot of diversity. If you go look at Endur's manual, and he talks about a plant. This is the type of plant that gets a plant breeder excited because there's amazing diversity, and it flowers in a year. Right, so if you push it, you can get these plants to flower and determine their traits in a year. So those are great things for a plant breeder. Also, I'm impatient, so I like to have to flower quickly. So I'm going to run through some of these. Here's Fiji. This is on the smaller side. It's a fairly small plant. Well, this is what we call that anemone type. Cross it with white chiffon, so it lacks that eye spot. And this is more of a semi-double kind of type here. And this is sort of what we get. So this, we have more petaloid stamens that are larger in our hybrid here than we did in Fiji. It's very reminiscent of Fiji. I don't think it's a self-pollen, self-seedling, but it's always possible. Nothing's out of the realm of possibility. But this is the same cross, and this is what we get. So every time I think we've got it figured out, so we cross them, a white with a little bit of pink in it, cross it with a white. Well, I would expect that you're going to get a white, and I would expect that you're going to have an eye spot. Well, we do have the eye spot. But this is my favorite Althea seedling that we've grown so far. This thing is I love the size of these petaloid stamens. They're larger than most of the anemone or double flower types. I'm really excited on that. But it really, I have no idea what's going on with the genetics. Blue chiffon is a really good blue, sort of semi-double anemone type, crossed with Fiji. And again, we get more of a pinky lavender. This guy, though, the petaloid stamens come all the way up the staminal column. I wish I had a side perspective of this because there are no anthers. It produces no pollen. Okay? And it actually, that gives it a little bit of a pyramidal sort of Christmas tree conical shape. It's a really interesting flower. This one's obviously a little beat up. Blue chiffon again, we cross with Little Kim. Little Kim is, as the name suggests, it's quite small. A lot of growers have told me it's too slow growing for them. I have to, it's a little bit confounding when growers say, I need a dwarf. Well, dwarfs, they grow slow, right? And then you give them a dwarf, and they say, well, not that slow. So anyway, I'm not talking about growth. Anyway, so the flower of this guy is definitely small. And again, we cross a blue with a white. We get sort of a lavender pink. And you can see there's just a couple of petaloid stamens down here. at the base. But what's really fascinating about this, pink giant is the largest flowering Althea that I've got. There may be some large ones out there, but it's the largest one that I know of. And this is that hybrid that I just showed you. And again, I don't understand how you cross basically a modest or moderate sized cultivar with a small flowered size, and then you get, you know, this gigantic. I mean, it's not like anyway, it's not dinner plate, but it's pretty good size. So anyway, we're pretty excited about that cultivar as well. So what do we think we know from all this so far? Is that white does appear to be recessive. This is not too shocking. This is generally what we'd expect out of white flower would be recessive. Petal count, so the number of petaloid stamens that we have, it does appear to be multiple alleles or genes, and they do appear to be recessive. Right? So that one still, I'm not completely sold on that. Eye spot does appear to be dominant. That one's the only one that I actually feel fairly confident in making that assertion. It does appear to be a single or very few genes, and it does appear to be that eye spot is dominant to no eye spot, which stands to reason there are more cultivars out there that have an eye spot than don't have an eye spot. Okay, so this is, and I don't, there will not be a quiz on this. I couldn't grade it. Now this is my graduate student Jason Ladier. I'm sure many of you know. He's an NC State boy. Possible flower color inheritance patterns. This is using the single locus model. Anyway, this Althea, they're tetraploids. It makes the genetics quite complex. And so this is how I'm just blaming it on complex genetics. That's why we don't have it all figured out. But I do want to talk for just a minute more about the U.S. National Arboretum cultivars that are out there. So Diana, which is a pure white, no eye spots. Minerva, Helene, and Aphrodite. Okay, so these were released by Dr. Egoff, who he was a very accomplished cytogeneticist. So he did a lot of looking at chromosomes, did a lot of polio manipulation, and so these plants. Quickly I'll go through. So to go back to the fertility thing. So we got these cultivars in, and I wanted to, for myself, I'm not going to just take a single reviewer's word for it. I wanted to put some numbers on how fertile are they. So we started out with 9 cultivars. We made a bunch of crosses. And you can see, so Aphrodite had 10 seedlings per flower, per pollinated flower. Diana certainly was fertile. It wasn't super fertile. Minerva was fairly fertile as well. The only cultivar out of all of these that was, had significantly reduced fertility was that pink giant that I showed you a little bit ago. You may maybe know this as Flogi or Flogii is the cultivar name that you also see out there with pink giant. So pink giant has reduced fertility. Note that. And that was as a female. Same thing as a male. Pink giant is the only one that really has reduced fertility as a male. Incidentally, Lucy, the reason why it has 0 is it's a full double. Doesn't produce any pollen, so you can't use it as a male. So anyway, again, that pink giant is standing out as being very low in fertility. So to complement this, we ran flow cytometry. This is a very cool way of quickly and accurately figuring out how much DNA is in a plant cell, right? So we ran these all through the flow cytometer, and of course at this point we've got more than our 9. And again, Althea is a tetraploid. So all of these are tetraploids except for pink giant, that one that had the reduced fertility. It also had the great big flower. So it's that I feel confident in stating that wherever you get pink giant, it's going to be a hexaploid. Incidentally, there's Aphrodite, Diana, Minerva. I think don't have Helene in here. At any rate, 4x, 4x, and 4x. So, what is going on? Incidentally, Jason also did to complement the flow cytometry work, which is very accurate, but nothing really takes the place of getting in and looking and counting the number of chromosomes. Diana has 80 chromosomes. It makes it a tetraploid. Okay, so what is going on with these things? So they were reduced. They were marketed and released as having basically no seed set. So I don't know what's going on. Why they appear to not have any seed set, if they were isolated where they were tested for an extended period of time at the National Arboretum, if they were planted in an isolation block with other plants that didn't produce much pollen, if there were no pollinators working. It's really confounding. But what I do know is if you read the releases from Dr. Egolf, it's sort of unclear on a number of fronts what actually happened. But what he does report is that he crossed these cultivars, right, or these seedlings, with an 8X, or with a what he said was a tetraploid form of William R. Smith. Well, so I throw that out because it's not a tetraploid. If he, he started with tetraploids. If he did in fact successfully double William R. Smith, it was actually an octoploid. And then if he back-crossed with all of these other seedlings over here, it actually would have been a 6X population. And if that's what he did, it does make sense because we've seen with pink giant that the fertility is greatly reduced. All right, so that actually does make sense. But I don't understand what happened between then and now, between when the plants were evaluated and what happened now. We've gone back to the National Arboretum and we've got cuttings from as close to the original plants as we can. On a couple of them, I think they are the very original plants, and they are tetraploid. So it's sort of a good news, bad news story for me as a plant breeder. The good news is ploidy manipulation is an effective way to reduce the fertility in Althea. The bad news, potential bad news, is it may not be stable. So that's one possibility, is that these plants through serial propagation may have reverted. Now, okay, if it's serial propagation, that's one thing, but why the plants that are at the National Arboretum are still tetraploids is beyond me. I don't understand it. So again, further evidence, I keep saying this, but we don't have it all figured out. By the end, I'm going to convince you of two things. One, I don't know very much, and I'm from North Carolina. So Katsura tree, moving on. So I love to talk about Althea, but another project we're working on is Katsura tree. Denny Werner gave me the inspiration for Katsura tree because there are two weeping cultivars, Morioka weeping and Amazing Grace, and there's a red foliaged form called Red Fox. That may sound familiar with Cercis, right? So anyway, in Katsura tree, I decided we're going to develop a red weeper. Katsura tree is a dioecious species, and it just so happened Mark actually is the one who helped inform me that Red Fox is in fact a female. And I knew just from my own experience with the weepers that both of them are male. So I thought this is going to be the easiest breeding project ever. I'm going to stick them in the ground. I'm going to walk away. I'm going to go relax and have a cold beverage. And they're going to pollinate themselves because you don't have to emasculate. You don't have to do anything. They're wind pollinated. It produces a load of pollen. Unfortunately, Red Fox is really reticent to flower. This one is at Heritage Seedlings down the road from me in the Willamette Valley. And this one, Eric Hammond said this one's been in the ground for about 7.5, 8 years. Well, now at this point, 9 years, and it's still never flowered. So I said, okay, we can't wait, that long. So we started getting all science-y, and we're taking, GA, gibberellic acid, and we're going out and we're spraying them in the summer trying to induce flowering, and that worked like a goose egg. No flowers. But my research assistant went across the street to Blue Heron Farms, and they had about 200-300 Red Fox in 7 or 10 gallon cans. She went through all of these plants meticulously and found one plant that had flowers on it. And Mark can tell you how big the flowers of Red Fox are. The female, they're 8-petalless. It's basically, all it is a little a pistil sticking out. It's like a little tiny hair. They're very inconspicuous, very hard to see. So Mara got a big gold star. She found the plant, and lo and behold, so this is basically what the flower looks like. It's essentially, it's nothing. This is a fertilized pod that's developing. Ovary is swelling here, but that's what the flower looks like. So she's good. We're going to keep her around. So this is Red Fox. We did get seed set. We got lots and lots of seed. These are, I actually cleaned these myself. So this is Red Fox by Morioka weeping. Got lots of seed. And then we had lots of Pythium when my seedlings germinated. So I lost, these are so sad looking. But I did have 12 seedlings that survived from the first year of crosses. We have planted them out. So quickly, I think these are going to be red. They have very distinctly red petioles. If you look back here, this is Red Fox inside the greenhouse. And this is what Red Fox looks like inside the greenhouse. Okay? It expresses very little or no of that, none of the purple-red pigment, although you can see it has red petioles, but very little purple in the foliage. But if you put it outside, this is what it looks like. All right, so inside the greenhouse, that's why I think my seedlings, you can see they have a distinctly red petiole, but the foliage isn't real red. It's got a little bit of red at the top. I think they're actually going to be red because I stuck them outside, and immediately we started getting some of the expression of the purple or red pigment. So we've got them planted out in the field. I think they're going to be red, and I'm actually really excited, even though they're not red weepers. The problem with red foxes, as I showed you that plant that's out in the mounds, it's just a little shrub. What would be really fantastic is if we had a nice, majestic, fast-growing, true shade tree form of Katsura tree that had red leaves. I think that in and of itself would be a massive breakthrough, and I think we've got it here, but you know, I've thought a lot of things in my life. But 12 plants, any plant breeder will tell you, any plant breeder worth their salt will tell you, it's a numbers game. So if you really want to get the trait, you don't grow 12 plants, okay? So anyway, we mowed those crosses again, and this year we used bag mix, and we very carefully controlled the pythium. We had no pythium. These plants all are growing out. We got actually both. So the previous year we only had Morioka weeping. This year we got some amazing grace crosses. Grow them out there now about chest height, and they're showing quite a bit of the pigment here. So again, I think what we've got is weeping is recessive. It is obviously recessive. None of those plants are weeping, and red foliage is probably dominant. I can't say for sure whether it is or not, but just to quickly review, I mean, hopefully all of you are familiar with the story, but there's Denny Werner again with Ruby Falls. And so what he did is he said, hey, there's a weeper out there, there's Cubby, and there's a purple foliage forest pansy. Why don't we develop a purple foliage weeper? Here's a shot. I was out touring his greenhouse with him many years ago, and so this is just a shot of the bench. I was actually talking with Tom about this. I thought my memory doesn't serve me. I thought that looking back on it, that Denny said that red foliage was dominant in Cercis, but then talking to Tom Rainey, he said that he thinks that actually red foliage was recessive. But regardless, that gets the blood racing. I mean, think, if I was still working in peanut genetics, I'd be in Lewiston looking at, a field of peanuts. Instead, I get to hang out with people who do this kind of thing. So another example, this one's hot off the press. This is Chocolate Fountain albizia, developed by Dr. Rainey, and really just a breakthrough. Also has some disease resistance, I believe, but combines the weeping habit with purple foliage, and in this case, I'm sure the weeping trait that you see here, and this is a J.C. Rawson photo, probably one of Mark's, I imagine. But the weeper is recessive, the weeping trait is recessive, and the purple trait is recessive. So you cross that first generation, and what do you get is an upright tree that's green. Right? So you actually have to go to the next generation. So it's heterozygous, and it's expressing those dominant traits are masking the recessive trait. So you have to go to the next generation to recover, 1/16 of the plants that are going to have homozygous recessive for both of those traits. So that's the sort of thing that we're trying to do with Cercidophyllum. All right, moving on to Philadelphus. So this is a couple of quotes. The only redeeming character is fragrance. Anybody know who this is from? If they're bad-mouthing a plant, I've never seen anybody who loves plants so much badmouth plants so much. Yeah, yeah, right. So the purple sepal types initiated pronounced heart palpitations. So even Dr. Durr got very excited about the purple sepal types. I do too. But then you talk to Tim Wood. Tim Wood and Dale Deppy were out there from Spring Meadow Nurseries, and I said, you're going to need, this is what Tim told me, he said, you're going to need to do a lot more than have a purple sepal to make mock orange a big seller again. I have to agree. But I said, what about a double-flowered pink, purple sepal that has the fragrance of what I've got? Okay. But the thing about Philadelphus is it's an easy plant to grow. Anybody can grow Philadelphus. It's known. Even if you're not crazy about them, everybody knows Philadelphus. And there's a lot of variability. There's a lot of untapped variability in the genus. All right. And so I wanted to start with fragrance and inflorescence architecture. I was down with Neil Bell at Santa Cruz Arboretum, and we were collecting Proteaceae stuff, Grevillea, and all kinds of weird xeric type plants. And we were in the back of the Arboretum going to treat these cuttings to bring them back to Oregon. And I was about 90 feet away, 100 feet away from this plant. It was about 4 or 5 feet tall, big, rangy, spreading thing, but it smelled like grape soda, and I could smell it from a mile away. So I said, I have to get some cuttings of this thing. I've never smelled a mock orange quite like it. Just really, really incredible. But you can see it's a solitary flower. It doesn't have really great habit, and cold hardiness may be a little bit iffy, although it's done fine in the Willamette Valley. We're not that cold. But I thought, if I could get something that had at least somewhat of an branched inflorescence with this fragrance and improved the form a little bit, you might really have something. So that's where I started. I've since included a number of other cultivars. So miniature snowflake is actually a good-looking garden plant, right? It's got these double flowers, but how's the fragrance on miniature snowflake? Nothing, right? Why are you going to grow a mock orange if it's not fragrant? That's just crazy to me. Anyway, Snow White, another one. I don't have gigantic hands, but that's a pretty large flower on a mock orange. Again, if I could get a branched inflorescence that has my fragrance of that improved Philadelphus mexicanus, with something like that, I think we'd be doing good. And then there's this Philadelphus texensis variety Corianus. This was from a plant we got from Dancing Oaks Nursery. Leonard had this really great plantsman, but a glossy leaf. So this is a different way of thinking about a mock orange. I've never seen a mock orange that had glossy leaves before I saw this guy. Really tiny leaves. They're only less than about a ½ inch long, and it's a little tiny plant. But I think this is going to be a real game changer for our program. We started with humble beginnings. So this is my mexicanus. I pollinated every flower that came on that plant. I actually did make all of these crosses. And this is Philadelphus lewisii blizzard, our native mock orange, the West Coast. And then this I hadn't keyed this thing out, but the inflorescence is, it's 8 inches long, has up to like 16 flowers on it. It's really incredible. So we started with just these 3 as our parents. We've now expanded to about 12 or 13 parents. And so we know some things, and we continue throwing lots of new traits and lots of different parents in. So I'm going to continue confounding it so I know nothing. But what we do know, and going back to earlier when I talked about the azalea dendrons, in direct opposition to the azalea dendrons where fragrance fell out in the F1s, the fragrance, this smells exactly like mexicanus. It is just like opening up a can of grape soda. If it was national, I want to call it knee-high because they're low, compact. It smells like grape knee-high, but I don't think everybody knows what knee-high is. Anyway, fragrance is coming through. There's not a clear ratio yet, but fragrance is coming through, and I can't tell. I've never been more excited about a group of plants than when these started flowering, and they actually smelled like grape soda. But the architecture on this group of F1s, we have about 500 seedlings out there, and I've just been really impressed at how fast the architecture of some of these has improved. Going from a rangy, sort of mutt-looking plant, this is a nice upright fastigiate plant. And then there's this that I'm about as excited about this as I am any of our seedlings that are out there. This is that cross with the really long extended inflorescence in mexicanus, and it smells exactly like mexicanus, and it has branched inflorescence. It has really good form and habit. I will admit, as Dr. Bilderback would say, you could probably throw a dead cat or two through there, but this is me beating this plant up. We're not treating these plants very well. We do have irrigation, but you give it to a skilled grower, and I imagine it would be a lot different. So here's an up-close shot. Every inflorescence on this plant had 3 flowers, and so of course the terminal flower opens first, and then these laterals open, which extends the bloom time. So it's a pretty long bloom time, amazing fragrance, and really nice architecture. And like I said, so what we know about the genes here is that that branching inflorescence, it is heritable. So we've increased the flower number in these inflorescences. But again, there's no clear dominance pattern that's emerged yet. So this was really unexpected. We are using Tenua folius, and there's 2 or 3 species. I need to go figure out what we are using. But none of those parents that actually have a purple calyx was included in these crosses. This is just some random happy accident. Don't know why, where it came from, but we got this calyx had pigment. It had a branched inflorescence, as you can see, 4, 5, 6 flowers per inflorescence. Not every one in this case, so there are some solitary flowers. And it did have the strong, strong, strong grape soda flavor. Really, really excited about this. This is one, I put flags as I like the plant, as I continue liking the plant throughout the seasons. I'll continue putting flags down. So a plant by the end of the year that has five flags in it, that's winter. This one, for form, if it wasn't flowering, I didn't select this plant. But nonetheless, it did have a flag in it, so we're going to advance it to the next generation because these are great traits. It's not worth the burn pile. Cotoneaster. This is one, and I think this is my last big crop I'm going to talk about, and I'll run through it pretty quickly, but I think it's really important. It's really fallen out of favor. There used to be zillions of Cotoneasters that were grown, and I think there's a lot of potential in this genus. People still ask me, why the heck are you working on that? But okay, for those of you not familiar, it is in Rosaceae. It's in the apple subfamily. And in the genus, there are 2 subgenera. One is subgenus Cotoneaster. It's got these little tiny sort of cloistered flowers. They never really open up, really sticky, really a pain in the butt to cross with. And then Chenopedilum is the other subgenus, and they're lovely to work with. And so this genus, subgenus, is things like Damari, Coral Beauty, those things that you're more probably familiar with. 11 sections though, 400 species, all kinds of diversity that nobody ever thinks about, It's like Skogholm and September Glory and Coral Beauty. There's more out there. There's more in the world. Life's too short for boring Cotoneasters. So again, this genus, it flowers sporadically over a long period of time. Maybe it creates an increased risk to fire blight, which I think is one of the reasons why it's fallen out of favor. This other subgenus, they flower all at once, maybe gives you a little bit more oomph. Lots of flowers, tend to have lots of flowers open at the same time. Short duration. A couple of interesting facts, I don't want to bore you, but 70% of the genus are actually tetraploids, right? And 15% are triploids, and then there's about 10% that are diploids. Why does that matter? Who cares? The thing is, Apomixis is really prevalent in the apple subfamily in Maloideae. Right? And so Apomixis is the production of asexual seed, and in the case of Cotoneaster, it actually contributes to its weediness. Well, we don't think about Cotoneaster being weedy out here on the East Coast, but in California and in coastal regions in southern Oregon, it actually is a bit of an issue. So it is common in Maloideae, and in some genera, it's been shown to be dominant. Apomixis has. That's not good for us. But the polyploids in Cotoneaster are obligate apomix. So if you go back, 90% of the species are polyploids, so that means 90% are obligate apomix. That's not good for breeding. If they're producing asexual seed, what we like is to have sexual seed and genetic recombination. So apomixis is not good. But luckily, 10% are diploids, and they have been proven to be sexual, right? So they produce sexual seed. But again, just to make the pitch for Cotoneaster, multiple season interest. They're insanely tough. I'll show you a trial that we did, and for 3 years in the Willamette Valley with no irrigation. Well, you think we have all this rain, but basically from about July until October, it does not rain in the Willamette Valley. And these things are basically growing in concrete at that point, and they just do fine. But there's a diversity of habits, flowers, foliage, fruit, that's really incredible. Again, did I mention that they're tough? So don't dump on Cotoneaster. Maybe prosecuted. But I mean, I'm not super crazy about, basically this looks like green concrete to me. I mean, this heavily sheared, and this is Coral Beauty. But man, are they tough, and they are useful. And anybody know where this is? My part of the world? Bouchard, absolutely, yeah. There's a waterfall and fountains over here. I think this is lovely. I mean, it softens this edge. Lots of these species cascade. And there's a rock spray, a little bit unkempt, perhaps for your taste. But I mean, this is just lovely. It softens these edges of the rock while not hiding it. And so we put in, this is 49 or 50 species of Cotoneaster, and we just put them in the ground, and we just watched them for a few years. And again, a lot of these species came through -4 degrees Fahrenheit, which is pretty cold, and they weren't irrigated for 3 years. We irrigated the very first year, so don't call me a liar, there is irrigation. We did establish them. But just insanely tough, and the diversity is fantastic. Fire blight is a problem, though. I think it obviously has contributed to the reduction of Cotoneaster in the landscape. There are actually bans on Cotoneasters in Europe. They're worried about it being a source of inoculum on the pear and the apple industry. And in Finland, their plants and live pollen have even been banned. Okay? In breeding for fire blight resistance in Cotoneaster, there's basically been mainly observational studies with very little inoculation, but it has been proven that the diploids do segregate for fire blight resistance. That basically, all that tells me is that it's possible to breed for fire blight resistance in Cotoneaster. The problem, again, this apomixis-assisted invasiveness. So if you've got an apomix, it doesn't matter if it's cold, wet, and the pollinators aren't flying, they're still going to produce seed. So there's loads of seed produced, and again, it's an issue in coastal areas in California, Oregon, and Washington. And again, it's a big issue in England and Wales. A number of species have escaped cultivation. We've got actually 36 that have escaped cultivation and naturalized and become identified as invasive in the U.S. And around the world. There's a number of places. They're survivors. Things that make really, really good landscape plants tend to make kind of good invaders. Okay. So what we're doing at OSU, we're breeding for disease resistance, we're breeding for sterility to prevent this kind of thing from happening, and we want aesthetics and performance. Everybody, all of the nurserymen that I have come through, they always put stakes or flags in these little tiny low growers, so that's what they're doing. So to identify sources of fire blight resistance, we did a couple-year study. We had 50 species in there. We did it in a greenhouse. This is Joseph Rothluchner, my first master student, who is now tree improvement specialist at the Morton Arboretum. So he did all this work on fire blight resistant screening in Cotoneaster. We dip the scissors in the inoculum. We then bisect the young leaves, and it's a really effective way of inoculating them with the fire blight. So in 2011, we had 8 species that had 5% or less fire blight. So this is a completely susceptible plant. This is a totally resistant. You can see this is where we inoculated, and it keeps growing its full head off. 5 species that showed no disease symptoms, and then there was a fair amount of susceptibility. The next year, overall, there was a reduction in severity, but we still had a number of species that were excited had 0 resistance. Not going to talk about that. So basically, Coral Beauty is a diploid. It produces sexual seed. So that is our female. We cross everything with Coral Beauty. And I'm just going to talk about 2. So Splendens had no disease, and Divaricatus was actually susceptible. Okay? So we went in and we made crosses, and this is in subgenus Chenopedilum. This is Coral Beauty. Again, this is an emasculated flower here. You can see the pistil. There Joe's in the process of emasculating, quite tedious. So we crossed Coral Beauty by Cotoneaster splendens, and we've got these what are clearly hybrids. You can see the foliage is actually more reminiscent of the male parent. So, and we confirmed that these are triploids. And here's our process of going through. So we make these crosses. We can confirm that they're hybrids using flow cytometry. We propagate them to make multiple reps. We inoculate them to confirm that they have fire blight resistance. We then put them in our own field trials. We then take them out to nurseries under MTAs, and hopefully we release them. If they're not what we want, we go back and make more controlled crosses, start the process over again. Okay? I'm going to show you 2 hybrids that I'm most excited about. One is a cross between Coral Beauty and Splendens. I think this is a big improvement. It is a much better form than Coral Beauty, and it shows 0 disease susceptibility in our trials. So I'm very excited. This is that plant after being propagated, so pretty excited. This is the other one that I'm going to show you in about 2 seconds. I'm pretty excited about that guy too. This one is a cross between Coral Beauty and Divaricatus. Again, it is a triploid that shows no disease symptoms after inoculating in the glass house. This thing, it is so low growing. It has this really nice red new growth. It actually is, it makes me think about a gumbo azalea. Anyway, really, I think this might be our biggest hit so far. I'm just really excited by this. Monrovia is really excited. Some folks in France are going to be trying on it soon. Sorry, I know I had to ramble there to get through the end. I hope I didn't bore you too much. Please stay tuned. Come see us in Oregon. We still have American Elms out there, which is pretty exciting if you want to come see them. This is on campus at Oregon State. We do have a lovely campus. Come see us anytime. This is my F1 Cooper, and this is another just happy accident. I can't believe I was still able to find this photo, but again, I was just walking around the JCRA, which is my home garden, and saw this little kid. And we are, not to sound too corny, but I mean, this is who we're breeding plants for, and we are trying to plan and plant for a better world. And that's it. Thank you very much for your attention. Over like 30 minutes,
That's
but
okay.
maybe if there are any questions.
Got time for a couple questions?
Yes, sir.
On that hibiscus, your hybrid one, have you, that's close to how much you're on that one? It has the big flowers, is that,
So that hybrid that has the same size as the pink giant, it's actually a tetraploid. Well, I shouldn't say that. Because we have 1,300 seedlings, we've only been doing flow cytometry so far on the hybrids with pink giant. So basically to confirm that we have pentaploids crossing our tetraploids with the hexaploids. It's a more manageable, there's only about 200 of those. But yeah, it's worth testing that guy, because it is definitely suggests that it's got a higher ploid level. Yes, ma'am?
What's a meristem?
Meristem is just the growing point. Yeah, it's just the growing point at the top of the stem.
You put the chemical on to make it tetraploid.
Yeah, and so you want to treat the smallest population of cells as possible. So that's why you saw in the hibiscus where it was at the cotyledon stage, we put a little droplet that's actually got a little agar in it with our antimitotic agent, and you would put it on a tiny, tiny meristem. Yeah, it's right at the growing point.
Did you say that the hibiscus cedocella was going to be commercialized, or a chance of that?
I
I
guess.
love that plant.
Yeah, I think it's fantastic. I can't let it go. So I don't have any claim on it. I was just a graduate student. When you're a graduate student, you sign a piece of paper that says, I don't do anything with any of these, you own everything. Actually, you do that as a professor too, but I can't let it go. I've still got cuttings in my greenhouse in Oregon, even though I can't do anything with it. I still show it to people and say, hey, wouldn't this be great? Why don't you talk to Dr. Reuter? I think he's still got the plant, but it's just sitting there. Incidentally, he's now the Alan Armitage Endowed Professor at the University of Georgia, and so he is the herbaceous breeder down there now. So there's always hope. I'm still hopeful that it'll be out there. But I think if folks actually took a look at the production, they'd be a lot happier with it than they are Jungle Red or Panama Red or any of those others. I think it's much better. But I'm very, very biased. Yes, sir.
On your list of genera that you're working with, you had Sarcophacoca, which includes a lot of good plants from North Carolina. But I thought I had heard or read or something that they were tough to work with breeding-wise. What are you doing with them? Maybe I'm wrong about that.
No, you're absolutely right. It's so if you in the literature, Sarcophacoca is reported to be an obligate apomec. That means it doesn't produce any sexual seed. But again, you can't. I think that's. I hate to throw somebody under the bus, but I think that's an error that was cited and then cited and then cited, because we've seen that they're a facultative apomec. So certainly, and I actually yesterday at Green and Growing, I showed some photos of twin seedlings. So you plant one seed and you got two seedlings that come up. Well, that's surefire. That's confirmation that it's an apomec, right? So I don't have time in my life to make a bunch of crosses that may or may not be sexual, right? So, but I wanted to increase the diversity and to just sort of screw them up. So we did a mutation breeding program, and so you can lots of different options for mutation breeding. We do both what we call physical mutagens, which are things like X-rays or gamma rays, as well as chemical mutagenesis. And so this was one of those cases where we used ethyl methane sulfonate. It's a point mutagen, really nasty stuff, and we've been really successful. We killed a lot of seedlings, which is what you have to do in a mutation breeding program, and we've got a really lovely chartreuse form. I even got Tony excited. So chartreuse, Sarcococa confusa. We've got about a half dozen little dwarf selections of confusa, and I was actually trying my initial thing. I wanted to knock out Apomixis and then set about making crosses. I've been entirely unsuccessful in developing, wholly sexual plants, but we've got some really exciting mutants. So, but it's slow growing. It's no hibiscus. So it's going to take a long time. I'm trying to get plants that are large enough to take cuttings at this point. So, but it's exciting work. It's an untapped market. I don't know if anybody's working with it right now. Anyone else?
Thanks, Ryan.
Thank you very much.